Welcome everyone. Assalamu alaikum. Today, we would like to start our journal club session. And we have with us today Dr. Shahnaij. He graduated from Dhaka University and worked for ICDDRB at around 6 years and received his Ph.D. from University of Kanazawa in Japan. And now he is working as a research fellow at Mayo Clinic. So welcome, Shahnaij. So please start your presentation. Thank you.
Good evening, everyone. It's a nice opportunity to grab and share some of my research that I did in Kanazawa University. So the overall study was to develop some kind of vaccine against the malaria. So for this purpose, we are using adeno-associated virus and adenovirus. These two are the harmless virus, a pathogenic virus, and they did not cause much trouble if we inject in the human. So just to clarify, because lots of people have the doubt that you are treating something with the virus, and people have the fear about the virus. So why we are using this virus? So they are the apathogenic virus that we use this virus as a vehicle to transport our gene to the target cells. So that's the primary target of the virus. This is the graphical abstract of my overall presentation that I'm gonna be described here. So the main focus is the adeno-associated virus. So the adeno-associated virus, we incorporated the surface antigen of the malaria parasite, and we tried different type of immunization route, particularly intranasal, intravenous, and the intramuscular dosing. And then we checked the expression in the liver and in the muscles using the in vivo model, and all of these works are done on my mice model. Then we checked the protective efficacy, and we tried bunch of virus and bunch of transduction factors and how it's mediated and what is the efficacy, and we tried different efficacy and the protection model, and we found that the intravenous route, when we injected the adeno-associated virus serotype 8, so we found maximum delivery in the liver and as well as in the muscles. And then we checked the immunoresponse in the mice model, and we found that the sufficient amount of specific IgG and infiltration of the T cells in the active site, which is liver. So this is the quick background that the malaria is caused by the falciparum species. One of the, so the most virulent type is the Plasmodium falciparum. That's I work on. And in our lab, we have some project about the Vivex as well. So the complete cycle of mosquito is carried out in mosquito and human, and still we did not have any effective vaccine against the this disease because of different and various type of their life cycle, their complex mechanism, and they change their antigens in different stages. So at least there are 5 stages here it's categorized, but to found a good antigen to make a vaccine, that's also a challenging. So for this purpose, we use the transmission, the first stage. At that stage, after the mosquito bite, the parasite is injected, and then through the blood vessel, it reaches to the liver. At that stage, we found that the circumsporozoid surface protein is abundantly expressed, so we target that one. And our aim is to make some efficient vaccine against malaria, which can induce the strong humoral and the cellular immunity. And nevertheless, safety and cost effectiveness is also concerned, but not discussed in my research. So what we are trying to induce some neutralizing agent and some CD8 mediated response that can clear the sporozoites before reaching the deadly stages. So if patients are reached to the blood stage of the malaria, then this vaccine will not gonna help. So this protein is a major surface protein, and the expression level and in the Western and as well as in term of RNA, it's abundant. And this is required for the entry in the liver cells. And previous studies was found that the immunity against the PFCSP protein is correlated with the protection. So our goal was to induce in such a level the humoral antibody and the cellular mediated response in such a level that can provide protection. So this is the quick background that in our lab, we use the most common prime and boost strategy. So in the priming stage, when we infect some animal or human with some antigen, so there are initial small response, but in this stage, a lot of the immunoresponse is kind of primed. So in the next stage, when we use the sufficient delay and then boost with the same antigen, then we found that large amount of immunoresponse, and then we use the malaria challenge with the sporozoite, and then we check the protective efficacy. In our lab, we tried baculovirus, adenovirus with the baculovirus with adenovirus. So we tried different permutation and combination of the prime boost using the adenovirus, adeno-associated virus, and baculovirus. And we found that the protective efficacy and the immunoresponse against the adenovirus is the maximum. So in my study, we plan to use this adenovirus as a priming agent. So here you can see what I described, that my study is the continuation of the last one, but I just quickly go through the overall thing, that adenovirus serotype 5, this vaccine is a better vehicle for the priming. And if we can include the adeno-associated virus as a booster, that can give a long-term transduction in and it can persist in the cell for a very long time. So as a result, we have the possibility to have the large immunoresponse for a very long term. So we tried the adeno-associated virus, for a boosting, and successfully we found that 80% protection in 2 experiments. So that is also consistent. So my study was to change the adeno-associated virus with the different serotype. Adeno-associated virus 1 have the tropism only for the muscles, but the adeno-associated virus serotype 8, which is more potent and rapidly delivered in whatever route we use. I tried the intravenous and the intermuscular route, and in each case, we found that the delivery with the AAV virus is the maximum. And it can persist. I checked in my study, it's up to 32 weeks. We can have the persistent expression of this protein with this virus. And the neutralizing agent against this capsid for the AAV is very rarely found. So this is the another reason why we use this adeno-associated virus serotype 8 instead of adeno-associated virus 1 because of the neutralizing antibody. Recent studies, they found that in Chinese population and also in the African population, the neutralizing antibody against the virus itself is very high, 40 to 70%. So it means that when we use this vaccine, so before reaching the sufficient amount of the antigen expression, they are being cleared by the natural antibody. Sorry. So in the clinical, in the clinical trials, this adeno-associated virus serotype 8 have the excellent profile. It's have very less cellular toxicity level, and they are well tolerated by the human as well. So my target was to construct the gene construction we tried. So to maximize the expression level, then we have to use less amount of vaccine. And we optimize the expression level of PFCSP using different immunization route, and we check the induction of the humoral and the cellular immunoresponse, and we compare with our previously designed AAV serotype 1. So this is the construction of my plasmid. We construct the plasmid, and then we make the virus, and then this is the functioning unit. So we use the CMB promoter that's consecutively express the protein, and we use the ladder and the ladder sequence and some sequences to facilitate the expression of the PFCSP. With this one, we incorporated the membrane anchor G protein. So the G protein allow this gene construct to express in the surface membrane of the hepatic cells or in the muscle cells, whatever. But the surface expression is going drastic. It become very well expressed potently and intensely in the surface membrane. So we incorporated this G protein that's also derived from the VSV virus. And the final part in this construction, we use the WPRE that stabilize the mRNA level, and therefore it's optimize the expression of the protein. So here we tested the Western blot, and we found that the expression of the AAV8 in two different cell lines. I use the HEC293 T cells and the HEP-A126 cell lines. So in both cases, we found that the expression of the AAV8. So one concern that why I use the AAV8 instead of AAV1, which have the highest expression uh, in the mammalian cell line. So I will share some of the data which indicates that the Western blot data of the mammalian cell lines did not and not properly describe the animal model studies. So this is another one. So as I told you before, that I used the BSG G protein. So therefore, you can see the the surface expression of the PFCSP protein is potently expressed in the surface membrane. So that's the target. So that's well done. So now our vaccines are ready for the testing in the animal model. So we use the bulb C mouse, and I tried the nasal drop, intramuscular, and the intravenous route. So the first two methods, the nasal drop and the intramuscular routes, are very safe, and it doesn't need much technical expertise to inject. However, intravenous route needs some technical expertise and have some concern. But in mice model, we can do and try and to translate the expression and overall things. So this is the summary of my study. I found that when we tried these two, I tried and compare these two vaccines, AAV1 and AAV8, and this time we use the reporter gene, which is luciferase gene. So the luciferase reacted, and it will generate some light emitting, and we captured it in the cool clamped, I forgot the name. So it's kind of, the camera is very sensitive to detect the small amount of light and the slight changes in it. So we found that the intranasal dose with the AV1 is maximum with the intranasal dose with the AV1, but not with the AV8. And in case of intramuscular and the intravenous injection, I found that the AV8 is the maximum. So it is consistent with the previous study. I'm going to share the study. So this is the study with the intranasal dose. So when we use the drop, so with the AV1, only the nasal cavity is being expressed, this one. And we also bring out the brain to check that they can go and pass to the brain, but it was not in the brain. So the slide is not attached here, but we checked the brain is not being affected by the virus AAV1. And with the AAV8, we did not see any expression in the detection limit. We tried the intramuscular route, and with the intramuscular route, you can see the high level of expression, not only in the muscles, but also in the liver. So most of my talk will be focused on the liver, because the malaria is infecting primarily in the liver first. And when liver is being sufficiently produce the meningoid, and then it will spread all over the body by the blood. So I use this intravenous route. So you can see the expression is maximum compared to the AV1. So they are very highly potent, like 25 to 48 times higher expression. Yeah, here I can take some question that is it translatable to use the reporter gene to the medical field, which is the surface antigen and how it's going to be react with these things. So yeah, always we can raise some question and make our study questionable, but yeah, therefore the science is moving with the translational experiment on the mice model. So my next,
Sorry to interrupt,
yeah,
Shahnaij. I have a question. So in vitro, when the antigen of when you culture in the hepatic cell, the expression level of AAV1 and compared with the AAV8 was the similar, or sometimes you see you told that the Western blot expression is higher in
yes.
AAV1. Why intravenously, when you inject intravenously, why you showed less with nothing?
It's better to go previous. Yeah, I will take question in between and during or end of the presentation. So just interrupt if you are not clear, so I can. It will be easier for me to understand that in what extent I'm moving faster or slower. So yeah, the question is that in mammalian cell line, that is less translatable for the medical purpose compared to the animal model, and the best one is the human. If we can try in the human, so that's the best one. But because of the biosafety and the ethical issue, we cannot use all the drugs directly in the human. So yeah, that's the basic things. And in the mammalian cell line, what we prove that with this construct we can able to express this protein in the cell surface. So that's the one use of this Western blot data and the IC data I shared. But there are a bunch of things that may interrupt that in the mammalian cell. This is open and the availability is very easy, and not always there are lots of factors that may include in the animal model. So sorry, I just could you please repeat the question again? So
I'm sorry, please continue. I will ask later again.
okay. So I checked the protective efficacy of the vaccine. So for this purpose, I use the uh, adenovirus for priming, and then we collected the sera after 6 weeks before before the booster dose. In the boosting dose, we use the adeno-associated virus serotype 1 and serotype 8. And then after 6 weeks, we collected the sera, and we did some other experiment to check the expression level of AAV8 or AAV1 in the liver as well. And then after 6 weeks, we challenge with the parasite. And after the challenge, we check the survival curve, the live and death event. So for the live and death event, we check the blood and the infected blood. If I check the thousand fields, and if I found that in 1,000 field I did not see any blood 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 cells, RBCs are infected with the blood stage of malaria, then I consider it as a negative. So this is the result from the survival curve. So with the this is the priming, and this is the booster with the intermuscular route. And for this one, the priming always in the intermuscular route. So you can see that with the intravenous route, I found 100% protection. And with the adeno-associated virus 1, either with muscular or intravenous route, I found similar level of protection. However, that is not my expectation. I thought that the protection with the AAV8 as a booster with the intramuscular, I will get a little higher, but this is the experiment we did several times, and we found that uh, it is 60% protection we can achieve at the maximum. So then we checked the humoral immunity. We collected the blood in 2-3 time points. And in the paper, I showed only the before boosting and before the challenge. So it means the humoral immunity, the immunoresponse after the priming and the boosting. And we found that all of them are sufficient enough to produce enough humoral immunoresponse. But when I try to correlate with the protection, it doesn't explain anything, because even though you can see the humoral immunoresponse in AV1 with IV challenge is less, but it provides the similar level of protection. And in case of AV8, either intermuscular intravenous route, they are sufficiently good humoral response, but the protection is not correlatable. So there might be advanced or some other mechanism working here that may explain. So we check among the infected mice and the protected mice, and we found that the humoral immunity in the protected and the infected mice is quite similar. So there is no significant difference between them. So here, the humoral immunoresponse is failed to correlate with the protection. So my next plan was to check the cellular immunoresponse. So for that purpose, before going to there, we collected the liver from the mice after the challenge, and we did the ICC experiment, and we found the PFCSP expression in the liver. This is after 32 weeks, and we found that still the expression of PFCSP antigen in the liver. And I did another experiment. I collected the liver and did the RNA and then cDNA, and you can see the expression is slightly different. So I can correlate with the protection level, but this study was not to see the protection. But yeah, that may be a reason that why the protection level in the IV, in the intravenous route is higher compared to the intermuscular route, which is 60% and 100%. So that's, I make the conclusion and reviewer accepted, but my sensei, my teacher, my supervisor did not convince with the idea. And he rejected the idea, saying that, no, you cannot say truly, because you need a lots of experiment to prove that one. So what we tried, we collected the liver and isolated the CD8 T cells, and we extensively analyze that which population and subpopulation of the CD8 T cells are being significantly different among them. So this is just a process how I collected. This is the effector memory T cells. That are the working member. They are the advanced military. They are weapon. They are involved in killing the infected cells or the cells which are expressing the PFCSP. And we also found the effector cells and the affected memory cells. Both are highly expressed in this group. So then I become very happy that, yeah, I found something that may be interesting in the scientific guys. They may be interested. Yeah, you found something good. So yeah, one of the achievement of our study is that a lots of group, they are trying to induce the CD8 T cells with the different type of regimen, the viral particles or nanoparticles, and lots of different factors. So I tried this one, AAV8, and I found that a good number of CD8 T cells, effector memory T cells, effector cells are highly induced and infiltrated in this liver cells. So that's the interesting point. And all the reviewers, I got unfortunately 5 reviewers. So all the reviewers appreciate this result, and they say that, yeah, this is something that may be translatable in the human studies. So with the increasing dose, you can see the splenic T cells are going down. So it means that the splenic T cells are generated, and then they are coming to the liver, and they make the population higher in the liver. And I checked the dose. So this dose, different doses, the 10 to the power 10 viral genome for the mouse is effective enough to induce the T cells. And the induced T cells, especially the effector and the memory effector T cells, were very high. So this is the experimental validation. So we found it, and this one, I just make one line on this one. So we use the tetramer. So this is the epitope, CD8 T epitopes. So it's indicate that the whatever I found, the CD8 T cells, they are not just present there, but they are also activated. Sometimes you can see that the infiltration and the loss of population surrounding the infection area, the CD8 T cells are highly abundant, but they are not working because of they are just recruited there, but they are not activated yet. So with this virus, with this viral DNA therapy, we not only recruit the CD8 T cells, but also able to activate the CD8 T cells. So that is one of the logic that therefore it is the infected hepatocytes or the expression of PFCSP in the liver are being cleared by the activated resident memory T cells. So one of the great phenomena of the resident T memory cells is that the cells reside in the tissue, and they are not escaping the liver, and they are moving around the liver. And whenever they encounter the infected cells, they kill it. So this is not my sorry, I have to go back. So this one I collected from some of the researchers. They are doing the same similar type of research in this field. So they found that the resident T memory cells, they are moving all over the liver, and whenever they encounter the infected cells, they kill it. So this is the kind of patrolling police to minimize the crime. So they are the active player inside the liver. So this is the very last slide of my experiments. So this is the subtype of the resident memory T cells, and you can see the induction of CXCR6 and KLRG1. So both these subpopulations of the specific resident memory T cells, they are antigenically a little different and diverse, but they work and in a similar fashion and in a similar extent. So this is my curiosity that which population are being induced and function here. So I found that both of them are induced and both of them are recruited there. So I can say that both of them are involved in the killing of the infected cells. So this is the take-home message from my study, that the AAV8 vector vaccines with the intravenous route that can express the transgene in a very higher level in the liver, and it can boost the humoral immunity as well as the cell-mediated immunoresponse. And with the standard dose we tested, it induced the CD8 T cells and the memory T cells. And the good thing is we found the 100% protection with this one. So this is the conclusion that this vaccine may go for the next level for the for the advanced study in this field. So people are interested in this kind of research to induce the CD8 T cells. So this is one reason that I found this study also interesting for me. And just to acknowledge all my co-workers who helped me to make my dream comes true, especially my supervisor, Yoshida. He have around 35 years of experience in the immunology and have a good, clear, sound knowledge in immunology. And some of our guys from Bangladesh, they are helping me a lot to carry it out and to have some fun time in Japan. So thank you very much, everybody.
Thank you very much, Shahnaij. It's an excellent presentation and excellent research work. So now I'd like to, means if anyone has any question, please unmute yourself and then ask a question.
Shahnaij bhai, I have a question.
Yeah.
I have actually, I think, several type of question. My first question is, you selected one species, Plasmodium falciparum, that's great. But there are several strains from the Plasmodium falciparum. What is your thought about protection against various strains? Did you try with multiple strain or you tried just one strain?
I tried the most abundant antigen from the falciparum, but yeah, it is true that we have the 1,000 types of different type of strain and substrain of the falciparum species. So particularly in the origin in Africa, it is too many. So, and it is difficult if we combine everything in one small vehicle. So it is our limitation. And to make the thing translatable, we use the most abundant strain.
Right.
So
Yeah.
the strain
This is
name is 3D7 if I really collected correctly.
right. Right. So this is my thought. This is the main challenge of developing any kind of vaccine due to the diversity within the one species. So if we could target one common antigen that's almost present in every strain. So that's why my question was about that. If your antigen, your targeted antigen was common among multiple strain or not.
Yeah,
Okay.
this is the similar type of questions are being asked to the influenza virus. People are making the virus. We are taking the vaccine shot every year, but the new emerging strains are being emerged, and they are making the thing worse.
Right.
So yeah, it is correct.
Okay, perfect. My next question is, you have shown that the correlation between the first dose versus booster in the humoral immunity, but you did not show the relationship within the CD8 positive cell if I did not miss something.
Oh yeah, it is. You do not miss anything. That's the limitation of my study, and uh, successfully we at that time we have the COVID at the full bloom.
Okay, I see.
So yeah, we have several study. We have several projects to carried out. A lots of question to be needed to be addressed. So we cannot do it, but yeah, we suggested some planning of the, but we are not able to because of the resource and the human resource and
Right,
availability of everybody in the university.
right. Okay, my sorry, I'm asking
Yeah,
you again.
truly we have three planning for that one. First one, we tried the anti-CD8 T cells. We will inject them to capture the CD8 T cells.
Okay.
So if we can capture, so we are expecting that the protection level will drop.
Right.
So that one is the first study. We make the dream, but it was dream. Yeah.
Right.
The second one is to use the knockout CD8 T cell knockout mouse. That's the second one. And the third one, people do, but we have the technical limitation on that one.
Right.
So the thing is, we plan to induce the CD8 T cells and extract all the CD8 T cells and then inject those CD8 T cells in the mouse liver, and when they are harvested, then we will try the protection study. So that will be a super brilliant idea, but we cannot.
All right.
I told you that's what.
All right.
Is this?
Moin, can I ask another question?
Yeah. Sure.
All right.
Sure.
So you have shown another data that was very interesting, that by increasing the dose, CD8 positive cell declined in the spleen population declined, and the hepatic population increased according to the dose. And you concluded that the spleen CD8 positive cell moved toward the liver. How did you confirm it can be due to like
yeah, I told that it may be go from spleen to the liver. That may be.
Okay.
Theoretically, the things happen that the spleen is the generator of the CD8 T cells. everybody will believe that one.
Mm-hmm.
But what factor make them express in different cell lines, and when they come to the liver, they are being restricted. So there are some play and interplay between the different type of markers. So they make those things happen in the animal model and as well as human. So there is another reason. I asked one of my friends that I found that declining population of spleen. So he suggested that this is not my suggestion. It is from the another immunology guy. He said that if you are infecting some virus and the virus is continuously producing the antigen. So for if some antigen is expressed for a very long time, so you can found the splenic population is decline over time. So this is the natural phenomena as well.
Mm-hmm. Yeah. Also, we did not check out other tissue because sometimes it happens. Spleen activate the CD8 positive cells and it is spread out throughout the body to make the greater protection. So number one, we cannot claim that these cells directly come from the spleen, number one, but yeah, I believe they came from the spleen. The second thing is this migration is very specific to the liver because we did not check muscle, we did not check other thing. Did you? Let's assume this migration is true.
Yeah.
The CD8 positive cell migrated from the spleen towards the liver. My question is, again, these specificities too good to be real?
Yeah, in terms of biological bodies, you can assume anything. One of the question that one is arise during the presentation in the Mayo Clinic, because they are the doctors and they have concern about the liver health and the toxicity of the health, right, because those cells are highly expressed in the liver. So the hepatic cells will be cleared out. So it may cause jaundice as well. So they suggest to use the to express in the muscles and check the expression level in the liver as well. So so far we found that that's not the case. To have the resident memory cells in the liver, I need something that is annoying in the liver. So therefore, it is kind of a smartness of, you can say, the brain of the immune response, that how they manage those things. There are lots of transcriptional switching inside, and they can sense that these cells are infected. So bring all things here, not to the other place. And there's another factor that sometimes you can see that in other cells also there are infiltration of CD8 T cells, but those cells are expressing PD-1 cells, the inhibitory, so in that case, they will receive the antigen, but they will do nothing. It is kind of cellular senescence. They are kind of, whatever you did, they just, okay, they will do nothing.
Right. Okay, my last question. So I'm not a vaccine expert, but immunology is my interest. That's why I'm asking. When you challenge with the infection after vaccination, do you use multiple dose, several dose? What if your stress test, the dose you are using in your stress test is something manageable with this kind of vaccine, but if you increase the dose of your stress test, your vaccine may fail? Did you use multiple doses for that or no?
I use multiple doses to found some effective dose.
Okay.
So that's one of them I showed in the induction of the CD8 T cells. Okay. So we found that the cutoff value is 10 power 10 viral genome per mouse is that
no, I'm not
Yeah.
talking about the virus. I'm talking about the falciparum, because we know this is the one of the important virulent factor, like how many organism is entering in our body,
Oh, yeah.
right?
Oh, yeah, yeah. I got
What?
the question wrong.
Yeah, yeah, yeah. So what if your vaccine is protective against for example, thousands of organism, but your vaccine is not protective against 10,000 organism? Is it possible?
Yeah, like when in our lab, when we found something protected, the mouse is
Okay.
being protected with the
Okay.
vaccination. So
Okay.
we do a lots of torture on the little animal, but
Right.
we just showed in the experiment, and due to the bioethics and animal welfare,
Mhm.
so we are not going to suggest anybody to challenge and re-challenge the falciparum dose again and again with the higher
Okay.
dose. But uh, in practical, in our lab, did those experiments, and those experiments are not successful. We just cut it out. But
Okay.
in some of the experiments, we found it is effective, that
Okay.
the multiple challenge and re-challenge, and we escalate the dose,
Mhm.
and they are still effective.
Hmm.
So that's good, and it is multiple challenge
Mhm.
is good for the immunology as well. If you are being infected, even with the small dose or the higher dose, that's good for maintaining the immune response.
Right. Okay. Thank you so much.
Yeah. Thank you.
Yeah, thank you very much, Shahnaij. So I think we should conclude our session.
Um,
Okay.
okay.
Ashish,
Mohan bhai,
I have very basic
Mohan
question.
bhai,
Yeah. Go for it.
You use several kind of mice or only the single type of mice?
I use single type of mice.
Okay, so
So
what
there
is
are some specific reason for this as well. So I use the BL6 mice,
mm-hmm,
not the black 6 mice.
mm-hmm,
So in the most of the study, they use the BL6 mice.
mm-hmm.
So one of the advantages of the bulb C mouse is that the immune response, the humoral immune response is very similar to the human.
Yeah, of
Very
course.
similar.
But the main thing is that if you work with CD8, it has some suppressor, it has some also some exhibit factor as well, like a TG8, TGF8, beta alpha,
Yeah,
gamma alpha.
correct.
So
Yeah,
if you suppress that kind of matter, you can see the product very well if you transgene the mice. So I don't know, in your research is a very outstanding research, but I think transgenic mice was used or not. So that was my question actually.
yeah. So yeah, one may use the BL6 mice. That's the second good choice to test. But one of the technical limitation from that angle is the IL-4 and IL-10. There are lots of cytokines are different in these two strains. I do believe that may have some role in the immunoresponse, and technical limitation we have that we use the CD8 epitopes that's only available for the uh, bulb C mouse. We did not have it for the black 6 mice. So that's the limitation we have. But yeah, if we can do a challenge in the protection, so that would be a good try.
I have just only one question again. So if we like in some cases we can do like the immunosuppressor, like we can suppress the immunization of the mice by doing the radiation, and
Hmm.
after that we can see the vaccine effectiveness. So did you do that kind of things in your research?
No, no,
Okay, okay.
no.
But
So
it's
just to make myself clear that the question is to uh, I did not get the question.
okay.
So
So like it's like that. If we want to see the vaccine, or if we want to see the effectiveness, sometimes we suppress the immunization power of the mice. Like if you use the BL6, there is no transgene, but you can do the suppression by the radiation, so that
uh,
the mice, after the radiation, the mice become suppressed about whole immunization. And when you push your target gene or target factor, you can see the clear effect, because sometimes it work or some factors can work. So I just curious
yeah,
that.
yeah. I understand. Yeah, like there are several branches. So I work just a very small focus part, and our lab is designed for that one. But a lots of question and lots of sides before translate some animal study to the human. So it will take another decade to understand everything and all of the components. Still, when we come to the human, so still we cannot correlate. And thousands of studies are
Yeah,
being tested and they're failed.
of course. Yeah, sure, sure. It's a very nice study. Thank you very much for your
Yeah,
presentation. Thank you.
thank you very much.
Mohan bhai, can I make a question? Can I?
Yeah, yeah, of course.
Okay. My name is Mahfouz. So I think it's very promising study. I have very like I want to know. Do you check the exhaustion marker of the CD8 T cells in the liver?
Exhaustion marker means
It means like the LAC3. I mean, you check your activation marker like CD69,
yeah, yeah, yeah.
some KRAG, but at the same time, if you check the exhaustion markers like LAC3 or PD1, as you said, like
Yeah, yeah,
PD1.
yeah.
So yeah. So did you check when you do the
Uh,
research?
no, but that one is a good option to check because in the cell there are lots of inhibitory responses. So that may be a potential role they may play. But one thing for sure, if activation markers are being expressed,
Mhm.
you will not found the inhibitory domains are being activated as well. Because the biological system is energy efficient way. So what this is overgeneralize. I overgeneralize the thing, but the thing is the biological system, it's uh, energy, they use the energy efficient way. So if something, this is from the literature review. It is some guy did some experiment and they found that if the inhibitory domain are being expressed, so the dead domains are not being expressed, like the PD1 and LAC3, and there are a bunch of things. So they are not being expressed,
Okay.
and vice versa. Vice versa. If the inhibitory domains are activated, so you will not found the activation marker on them. So that's the beauty of the nature.
Okay, I got it. Okay, so that is.
But
Yeah, I am thinking like if you make the ratio, then you may know that your activation marker is higher than your suppressive markers.
yeah, correct. That's the interesting field. And I did one interview during the postdoc. So the from the Purdue University, and they have the NIH collaboration with them. So they found the study very good, and they asked the same question to me. So uh, I just go to the old textbook, old school, and say the general biology of the system. But they are doing some awesome research on that one. So they collected the CD8 T cells, and they check the overall expression profile, like each and everything. They have one branch, they check only the immunodomains, and another branch, they are seeing the metabolic aspect of the CD8 T cells. So how they are different. So they check a lot of things. That one is the good option for me to go.
Okay, just this question is related to the translation, like how the research can be translated to human. So we know that in human, the adenovirus is most most of the humans are exposed to adenovirus, right?
Yeah.
So they may have the antibody. So when you, so how do you define your study in that case?
So defining, I admitted that's one of the limitation of my study, to use the adenovirus, particularly the serotype 5. So in the malaria endemic area, sometimes the reports are 70% to 90% people have the natural antibody against the virus.
Yeah.
So the alternative way people use, they are using the chimpanzee chimpanzee
right.
strains as AstraZeneca. They use the
the Pfizer and
strain.
they both use, right?
Yeah. So that's the good choice. But we did not have that luxury to use the chimpanzee strain of the adenovirus. But the Oxford, we have the our lab have the collaboration with them, but until or unless they found everything done, then they will allow us to use the chimpanzee strains. So there are some internal things you should know, that the politicians and
yeah, of course.
the researchers are thinking a lot of things in a similar way.
Right. But I think your research is very promising, and it has potentiality to make translation to human.
Yeah. So there are something that that's the with the presentation, people may conclude the thing, but I make them sure that we are not continuing, the lab is not continuing to study that one, because of the, as Masoom question, that did you try the elevated dose of the sporozoite and multiple challenge and re-challenge study? So we did it, and we found that declining protection level. So therefore we just throw the thing. But the good thing is that this is the published, and I got my PhD, therefore I can say anything.
But nice job, good job.
Yeah, but the adeno-associated virus 1, in our lab, we did the challenge and re-challenge study and the dose escalation study. So they are working very fine. They are very good. So I think initially they have the 80% protection level. So that's good.
Yeah.
And AAV1 may be a good thing. And maybe two paper already came from my lab. If you can go and search with the AAV1 with the PFCSP, you will find those 2 papers.
Okay, good job. That sounds good. Thank you.
Yeah.
Thank you very much, Shahnaij and everyone. So it's a long session and long question and answer. It's good.
Yeah, that's good for me. Yeah, I learned a lot of things. Yeah, and to continue this conversation so long, it's need questions.
Okay, thank you. So now, thank you for all. And I'd like to conclude this session. Bye.